Hi, welcome to Illinois Science Council. I'm Monica Metzler, ISC's founder and executive director, and I am very happy to welcome you to our program tonight on the human genome and consumer genetic testing. A few notes before I introduce our speaker. We have another great program coming up at the end of April when we're going to host a cognitive psychologist who's going to tell us about the science of happiness. So, definitely something to look forward to. On another happy note, we're very excited that vaccines are rolling out. So now we've added a vaccinated line of t shirts to our selection of science swag at our bonfire.com store. All proceeds from every sale support our mission of raising public understanding and appreciation of science. We are an independent 501c3 nonprofit, so we rely on you, our supporters, the wonderful people who understand the critical importance of science outreach. It's your contributions that allow us to bring great science programs to adult audiences. So thank you for the donations and visit bonfire.com and search for vaccinated shirts. And now, on to tonight's presenter, Dr. Jeffery Schloss. He has a PhD in cell biology from Carnegie Mellon University and most recently was a director in the Division of Genome Sciences at NIH. So, welcome, Dr. Schloss.
At NIH. So I should say I'm no longer at NIH. So this is but I'm gonna try to observe the sorts of things that a federal employee would have to observe. So I'm not going to plug anything or try to say anything really terrible about anything. But I'm going to start by sharing my screen, and we'll see how that works. Okay, you should be able to see it now.
Yep.
Okay, good. So yeah, the human genome, I have about 30 or 40 talks worth of material in here, which I promise you I won't do. But this is when I thought more about what I was supposed to talk about. I was wondering why I possibly agreed to it. It's a pretty broad scope. So I'm going to romp through a bunch of things. I'm going to put a little bit of since I was involved in some of it, I'll throw that in here and there just for personal interest. And if you don't care, then that's fine with me too. Yeah, Human Genome Project. And so my goal, what I'm trying to do today is for you to be a better, to try to provide some background information to help you be a better consumer of DNA information. And that's information about you, your family, and your friends. Now, I was real interested to see how many of you have already participated in this, some directly and some perhaps indirectly. And so I hope we'll have a really good discussion at the end about what your experiences have been, because I will say I have not participated in this myself. Well, I mean, on the technology and grant funding side, yes, but not, I haven't had myself genetically tested. So, you know, as you all know from much reading and listening to talks and so forth, all technologies have upsides and downsides, and I just want to help you walk into anything in genetic testing and DNA testing with your eyes open. Now, please, Monica, I know I'm not supposed to have a lot of slides with a lot of text. I have these 2 slides to romp quickly through some context for how we got to the Human Genome Project, and then in 2 slides, how we, before, during, and then at the end of the Human Genome Project, what we realized we needed to achieve. So obviously, we've known about heredity for hundreds of thousands of years. People have bred animals, people have domesticated plants. But in the mid-1800s, 1850-ish, was when Gregor Mendel did his experiments on peas and really sort of codified and collected statistical information on how genetic information is inherited. But it was until almost 100 years later that the DNA was understood to be the genetic material. In the early 1900s, there was lots of chemistry going on in biochemistry, and people were isolating proteins and carbohydrates and lipids and nucleic acids from cells, but trying to figure out what they all did was quite difficult. And in fact, people thought for many years that protein was the genetic material because it was complicated. Whereas, as we know, there are only 4 really simple structural building blocks to DNA. And how could you possibly encode information for inheritance in that? So in 1944, Oswald Avery discovered and published that DNA is the genetic material. And only a few years later, the structure of DNA was solved. And that actually revealed a lot about how DNA works, which I'll touch on in another slide, but not really go into that. That's a whole fascinating story by itself. The first biological molecules for which the sequencing was done was actually proteins in the 1950s. And in the 1960s, the first RNA sequence was done. But just look at these numbers. It took 5 people 3 years. They used a gram of material that was isolated from 140 kilograms of yeast. And they were able to sequence 76 bases, 76 nucleotides. In early 1973, a small part of a bacterial genome was sequenced, 24 bases. It took 2 years. So 2 years, 24 bases, on average, 1 base per month of sequencing. In 1976, things changed a lot. There were 2 methods published for sequencing of DNA. And one of those is still used pretty actively today for certain kinds of experiments and tests. But it's largely been superseded. Using that method in 1982, the first genome, a bacterial virus, was sequenced at 48,000 bases. In 1982, same year, there were 500,000 bases in GenBank, which is the genetic code, the DNA database at the National Institutes of Health. It's an international database. They're actually partners in Japan and Europe of GenBank. 5 years later, nearly 10 million. So the amount of sequencing clearly grew, and this was largely because of this 1976 publication of these methods. By 1987, that method had been automated in a machine that could generate 1,000 bases per day. So think about, 1 base per month, now 1,000 bases per day. And very shortly thereafter, the formal planning for a US federal program to sequence the human genome started. In 1990, the Human Genome Project in the US was funded. I want to be very clear, the Human Genome Project was an international project. England was roughly on the same pace as we were, the UK. And France also played a huge role in that. And then very quickly, other countries joined. So the Genome Project is international. The first bacterial genome was sequenced in 1995. Then yeast, E. coli, C. elegans, these were what we call model organisms. These are organisms that are used to study all sorts of interesting biology. And the Human Genome Project sequenced yeast, E. coli, and C. elegans as practice, basically, to successively larger genomes, figuring out how to organize the logistics of the process and the information. In 2001, a draft human genome was published. In 2003, the quote unquote finished was published. It still isn't finished, by the way, but we're very close. And in 2003, with the completion of the Human Genome Project, we said, look, this is really great, but we've got a lot more work to do. And so the office I was in at National Human Genome Research Institute launched this revolutionary sequencing technology. Now, I forgot to say one thing. I'm going to back up for just 1 second. This sounds to most of you on this call, on the Zoom, like really ancient history. But I saw a few faces that might actually, for which this might not be ancient history. The structure of DNA was published on my 2nd birthday. Okay, so. This is another talk. This is the talk I thought I was going to give, but I'm actually not. All I'm going to do with this slide is say, over a period of 15 years, starting in 2003, the National Human Genome Research Institute of the NIH, National Institutes of Health, invested about $220 million in developing new sequencing technologies. This money went to small companies, big companies, mostly to academics who had interesting ideas. $220 million is not a shitload of money. It's a lot of money. But if you consider that commercialization of each of these platforms, and you can sort of see the years in which these rolled out, was in the $50 to $100 to $150 million of private sector money that went into the development of each of these platforms. And almost everyone on this list received funding in one form or another from our program. The upshot of that was two things. In this first slide, you see how the costs came down. So that first one was commercialized in 2004. It takes a few years for once a device has been commercialized for it to actually get put into practical everyday use. So there's a little bit of a lag here, but you see how the shape, the slope of the curve changes dramatically in 2007. And that was when people stopped really routinely using the technology that had been used for the Human Genome Project and started using these next-gen sequencing devices. And you see what happened to the costs. We have a 5 order of magnitude reduction in cost of sequencing. So that was a pretty successful program. The other aspect of that was that near the end of the Human Genome Project, a human genome could be sequenced by running 100 machines for 3 months. Okay, that was the newest machines at that time. Today, one machine that's about the size of maybe of an apartment-sized washing machine, maybe small washing machine, can sequence many human genomes overnight. So we've come a very, very long way, which is why we can have a discussion about the clinical use of sequencing and the recreational use of sequencing, because it actually can be done for a reasonable cost. So they're the guys who discerned the structure of the DNA molecule, borrowing heavily from another investigator, as you're all aware, Rosalind Franklin. And so here's, what is this DNA stuff? This is a simple diagram, and it's showing you that there are 3 bits to a building block of DNA. And so a building block is called a nucleotide. It has a phosphate group, a sugar group, and this thing called a base. The way the DNA molecule assembles, there's this backbone of phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar. And sticking out off of them are these bases, the A, T, C, and G. So that's how that molecule is put together. One of the keys is, as we alluded to before, the information content is in the sequence of these nucleotides, or in the sequence of the base on the nucleotides. So AGGT means something different than CTAA. You just have to be able to read the code. The other interesting aspect of this is once you have the code on one strand, you also have the code on the other strand. This is a little bit of what I was alluding to by saying that a lot about how DNA works was elucidated when they figured out the structure. So A always pairs with T, and G always pairs with C. So if you have one strand, you also have the information content of the other strand. Okay, so that's a quick lesson in what's DNA. The other quick lesson is, how do you sequence DNA? How do you determine the sequence? You start by extracting the DNA from your source. And it could be, in this case, tissue. It could be a blood sample. It could be a saliva sample. And lots of things, all the living material has DNA in it. Then you do a bunch of magical things. And those magical things differ depending on what sequencing technology you're using. And again, I've got slides at the end, and we could talk about this later, but it's not core to what we're doing, to what we would be most interesting to talk about right now. So you do stuff with the DNA molecules. In this case, you're attaching little fragments of the DNA to a surface. You make many, many copies of each of them, so you get a signal out to your device. And then you do the sequencing reaction, and the device reads out the sequence, and that information flows directly from the device into a computer to put it all back together and then be able to do some analysis on it. We're talking about human genome sequencing today, but I don't want to lose track of the fact that we've sequenced lots of stuff. And this started, I referred to some of the model organisms that were being sequenced while the Human Genome Project was going on. I used that old 1976 method back when I was a graduate student and a postdoc and an assistant professor. where you get a few hundred bases a day and you're doing really well. I used the manual process because the machines were really expensive and only very specialized places could have them. But lots of things have been sequenced. We've learned a lot about all of these. Again, if you want to talk about some of that later, fine, but that's not where we're going. So today, there's one database that contains about 80,000 whole human genome sequences, and they represent 60 distinct human populations from all around the world. This is really important because as those of you who are doing ancestry or genealogy are learning, the sequences, there are differences in sequences of DNA from people around the world. And that's how you can determine what your ancestry is. In this database, and I should say that there are many, many more human genomes done. This is what's in a database that people can use for research because the donors of the samples consented for research use of their sequence information. So really important. So in this database of 80,000 whole genomes, there are 800 million variants. Now we're getting to the part of the talk that's where we want to go with it. And it's talking about these variants. So those are differences between the genomes of individuals in that database, in that 80,000. So how do you picture the variation in a genome? So this is an article that happened to be published this past week in the New York Times Magazine. It's called Tracking the COVID Code. John Gertner was the author. And I've just took this out. When you look at this online, unfortunately, you don't see this image, but the magazine has the image. And what I want to show you here, so first we'll look at this page, then we'll look at this page, and I'll show you why. So here's the first page, and this is the first half, if you will, of the sequence of the coronavirus. And what I want you to look at is, can you see there over here, there's a white letter, and here there's a white letter, and here there's a white letter, and here there are, I think, 8 white letters. So these are where this coronavirus is different from some other coronavirus. These are variants in the coronavirus genome. And then the next page is just some more of that, just showing you a few. So the impression I want you to get from this is, look how much DNA there is there, how many letters there are on those pages, and look how many of them are different. There are 34 changes in a nearly 30,000 nucleotide viral genome, which is 1 per 880 in a fraction. You differ from the person in the adjacent Zoom frame at about 1 per 1,000 nucleotides. So not very different. If you lined up your sequence with somebody else, it's gonna look about like that in terms of where you differ from each other. You differ, by the way, from a chimpanzee at about 1 in 100. So there are more differences. So in order to, if you get into this idea of DNA testing, and whether it's clinical or it's recreational, you're going to come across a lot of terminology, a lot of vocabulary. And I'm not going to go through this slide. Don't worry, Monica. I'm just going to, just a couple of words. So the main kind of DNA variants people talk about today is a SNP, a single nucleotide polymorphism. And SNP and SNV mean the same thing, single nucleotide variant, single nucleotide polymorphism. Those are the most common ones. And that's what's used, as I say, in clinical and in recreational testing these days. There's another kind of variant that's called a structural variant. Because of the kinds of sequencing methods that people have used, it's taken much longer to learn, to identify and learn about the function of these structural variants. But there are many, many of them. There are many kinds, as you can see here, and they're becoming more and more important because we have the tools to look at them now. This is where some of the different DNA sequencing methods differ from each other. It's how well they can determine structural variants. Again, a topic for another day or discussion later on. I just want to mention one other kind, which is the STR, the short tandem repeat. And this is what forensics has been based on for years. It's still using its DNA. It's looking at a repeat like CA, 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 and in different individuals, that'll be longer or shorter. And it's generally in non-function. It doesn't affect the function of what's being expressed by the DNA. It's strictly a genetic marker, but it differs in different people, and so it can be used in forensics. Okay, we're not going to come back to that. Some terminology. I can't actually see what's at the top of my screen because there's a little band from Zoom up there. So I just want to talk about some different aspects of DNA testing. So one of this DNA testing approaches, and particularly the first one you may have come across, some of you may have come across. So there's karyotyping, and I'll show you what that means in a moment. Then this forensics, which is used for crime scenes, and I just mentioned that. And then what's mostly used today in the clinic and in recreational genomics is genotyping and DNA sequencing. So we'll come back to talk about that a little bit. Who offers DNA testing? Obviously, we've been saying all along, clinical testing in the laboratory setting, and then there's direct to consumer, are the main offers, ways to get access to DNA testing. And then why? There's obviously information for health, and there's recreation or curiosity reasons for doing DNA testing. And I want to be clear, I'm not knocking any of this. It's different, and but there are a lot of actually shared considerations. So karyotyping. So the way most of the public might have been exposed to the idea of karyotyping is if you have heard of or had experience with amniocentesis, or subsequently chorionic villus sampling, which was a way to look for genetic abnormalities in a fetus. And the result of the test is a picture that looks like this. And what we're looking at here are the 23 chromosome pairs. Pairs, that is, if we look up here at chromosome 1, there's one copy of chromosome 1 that comes from your dad and one that comes from your mom, and a copy of chromosome 2 that comes from your mom and one that comes from your dad. We can't distinguish which one is which here, but using the lengths of the chromosomes and the banding patterns, people were able to, the scientist is able to line up and produce this picture. Now, this is actually coming from a cell that's going into cell division, and the chromosomes that are ordinarily stretched out in order to do their actual job of coding for stuff that you need in your cells condense when the cell goes into division and form structures that look like this. So what the previous picture was, actually somebody did this with a dividing cell, took a picture of it, printed the picture, and cut out each of the chromosomes and laid them next to each other to produce the karyotype. That's how it was done. And what you could, and so with newer technology, obviously now we know some information of sequence from most of the chromosomes, from all the chromosomes, and you can make little probes that bind to a specific sequence within a chromosome and put different colors on different sequences and do an experiment like this or like this, where you can actually paint the chromosomes with known sequences. And this gives you a much higher resolution view of whether anything is wrong, whether anything is different in these chromosomes than what's quote unquote normal. And so if you're looking for trisomy 21, that's relatively easy because there are 3 copies of chromosome 21. But you can see here, you could see a much more subtle difference. I don't know if anybody's doing this anymore, because now we've learned that we can collect, that maternal blood has a small amount of fetal DNA in it. And so from maternal blood, really non-invasive except a needle prick, you can sequence maternal blood and actually get the sequence of most of the fetus's genome. And so if you're looking for something, in particular, because you have a suspected genetic abnormality or genetic change, you can actually do that by sequencing today. And it's because we can sequence shitloads of DNA because you're sequencing mostly the mom's DNA, but there's a little bit of the fetal DNA in there. So as long as you can sequence a lot and throw away a part of the information, you can get that from our new sequencing technologies. Okay, so genotyping and sequencing. This is mostly what's used for the kinds of tests we're talking about. I'm going to try to keep an eye on my time here. I know I'm not doing great. There are several different flavors, if you will, of DNA sequencing. Whole genome sequencing, which is just what it sounds like. You sequence the entire genome. Exome sequencing, which is sequencing only the most important parts of the genome. Of course, if you only sequence those parts, they're what's important. And people are still doing exome sequencing, as I will show you in a moment because it's cheaper. It's less material to sequence, but in order to make the library, we call it a library that goes into the sequencing machine. In order to make the library, you have to do manipulations that mess up certain kinds of information. And then the other commonly done DNA sequencing is gene panels. And these are collections of genes that are relevant to a particular clinical condition. And so again, it allows you to do less sequencing, spend less money, makes your interpretation easier because you're only gathering the part of the information that's most relevant to what you think you're looking for. And then there's this other thing, it's not DNA sequencing per se, it's genotyping. And what it does is it looks at many, many, many interesting regions of the genome. And the people who design the genotype assay define what's interesting. The challenge here is that you have to know what you're looking for. What people do is they take these SNPs and represent them on a gene chip. If you've heard the term gene chip, it's what's used for genotyping. And a typical gene chip, I can't remember if I have this in a, yeah, so I can't see it because of the way my screen is laid out, but the cost decreases as you go down this slide. So that's one of the reasons people use this. A gene chip, the best gene chips have about 700,000 features on them. So you could look at 700,000 different regions in the genome. Each one might be 10 or 15 bases long. So you're only looking at a small fraction of the genome. What they do though, is they put the most common variants that occur in particular populations or across the world onto the gene chip. So it's a very quick way, and it's very cheap. I think they're $20 now to do a whole human genome genotype. So you can do gobs of them and get a lot of important genetic information. Okay, so what kinds of diseases can we get information about? I would sort of clump two major kinds of diseases. Other than infectious disease. I'm not talking here about infectious disease, but we have Mendelian disorders and complex diseases that are largely of genetic origin. Mendelian disorders are mostly rare diseases. A Mendelian disorder, one change of a change of a single nucleotide in the genome of that individual is enough to cause a disease. Okay, a single nucleotide change. These are individually rare diseases, and rare is defined as fewer than 1 per 200,000 population. So there aren't that many people with each rare disease, but there are almost 7,000 rare diseases. So you multiply that out, it's a large portion of the US population and of course the worldwide population that has some sort of a Mendelian disorder. And we can use sequencing. People started using exome sequencing and they actually were able to solve a lot of Mendelian disorders, that is identify the specific nucleotide. And that means you know what gene it's in. And then once you have that insight into that gene, you start to get biological insights that can help you develop therapies. And that's why it's important. The other major kind of disease is the complex diseases, or also known as common diseases. And these ones, this is why we have 80,000 or 100,000 or 200,000 human genomes. Because in order to study complex diseases, you have to compare large numbers of people who have the disease with the same number of people who don't have the disease and look for what's different about their genomes. The problem is these diseases, there may be a changes in 100 different genes or 200 different genes, that each of those changes has a small effect and they add up to the disease state. And these are things like diabetes and cardiovascular disease, they're metabolic diseases. It's really hard to see those small effects. So that's why you need large numbers of people and a really matched case and control in order to, in terms of the populations you're looking at, to find these changes. It's one of the reasons why we wanted better sequencing technology, was to be able to understand these diseases. Then you can also turn it around, and once you start to understand it, you can apply those to patients and see what's going on in their genome that might be contributing to their disease. Okay, this is just the sort of the most currently most important set of clinical gene tests. ACMG is the American College of Medical Genetics. They have currently defined 59 genes in which there are mutations that are known to cause a disorder and for which there's a clinical action that can help the patient. So these are good things to test for, because you can actually do something about it. Again, these are mostly cancer predispositions, cardiovascular and metabolic diseases. It affects a large portion of the population. If you get this test, they're going to give you certain information, and they're probably going to not give you certain information. Again, these are clinical tests. You're probably going to do this through your physician. You won't get most of this information through direct to consumer. Some of them may be giving you some of it, but if it's a variant that's known to be pathogenic, or a variant that is expected to be pathogenic, they will tell you that. They will tell you or your doctor, depends how you've set up your testing. When you get these tests, you are going to have to sign a consent. And part of that consent is going to be, what information do you want and what information do you not want? So one of the things that they will find when they do this test is variants of uncertain significance. That is, the variant occurs in you, it may have been seen in other people, but nobody really knows whether it has any medical effect. And so it's called a variant of uncertain significance. Some people would be so freaked out to know they have one of these, they do not want to know. Other people say, well, I want to know because then I can follow the literature and maybe I'm going to find out eventually either that will resolve with more research and we'll find out that that particular variant is kind of a random change in my DNA and it doesn't really have any effect. Or it could turn out that it does have an effect. The other really interesting part of this is incidental findings. So you do a gene test and you're looking for something related to your cardiovascular disease, and you find out that you have a disease that's likely to cause you to have a tumor. The people who are doing the tests have to have themselves protected in both directions. That is, people might want to know about that secondary finding, or people, or the clinician may say, I have to tell this person that they have this other disease that we can do something about. So this is where genetic testing and in the clinic particularly can get really tricky. So it's walk in with your eyes open, read that consent, because you want to know what you're getting back and not getting back when you get your genetic test done. This is a lot to sort out, and there are people who are called genetic counselors who can help you sort that out. We need one of the problems is most physicians do not have the training, either because of when they were trained or just because this information is coming out so fast that it's really hard to keep up with. A specialist like a genetic counselor, that's their job. And they're the ones who can help you figure out what you should do about genetic testing and what you might do about the results of your test. So direct to consumer. Most of the direct to consumer genetic testing, as far as I can tell, is being done by genotyping. It's not by sequencing. So it's looking at some fairly large number of SNPs. And again, there's information that can be gleaned from that. as I mentioned, the most dense chips have about 700,000 genomic locations represented on the chip. But think about it, if two people differ at 1 in every 1,000 bases, and you have 6 billion bases in your genome, then you have 6 million differences between those two people. So a gene chip is going to give you information on the most common of those variants. That is the variants that are found. A common variant is defined as it's present in at least 1% of the population. But the rare stuff, which could be significant, you're not going to see. The other thing I want to mention here is we always talk about 3 billion base pairs. You have 3 billion base pairs in the DNA you inherited from your dad. And you have 3 billion base pairs from the DNA in the DNA you inherited from your mom. All of that DNA matters. So if you're actually doing this right, you've got 6 billion base pairs to analyze. It's a lot of material to analyze. Okay, and then the increasing the amount of genomic data supports expanded ancestry and geographic location information. So the more genomes we sequence from the more parts of the world, the more of that kind of information we can put on a gene chip and therefore get that information back when you have a genotyping test. Now, obviously, you've got to sacrifice, you've only got 700,000 locations on the chip, you have to sacrifice something. But that the collection of more and more genomes from more and more populations allows us to get better information. So you can get a lot of information, and a lot of good information from direct-to-consumer testing. They differ in what they're selling. So obviously, you know, when you go to buy a car, you buy a car that serves your needs. Here, you do the same thing with DTC testing. And as the research improves, you're going to need updates because, so for example, they will base, and I think it's the reference genome here. Reference genome remains a challenge. When they're doing comparing your DNA to see what your ancestry is, they are looking at a reference genome that's at a set of reference genomes. And each of these references comes from a different population or a different geographic location in the world. That's how they're able to say, you know, you have 22% Eastern European. It's based on that reference. As we collect more and more information, we get better definition, better resolution on these different geographic locations, and the ancestry information will get better. And that's why sometimes people's ancestry information report changes. Their ancestry hasn't changed, the report has changed because there's better data on which to do the analysis. I skipped over this one point. The analytical validity of the tests today is pretty good. These people know how to run these tests. They know how to read them, and they know how to get a good computer analysis of it. That wasn't necessarily true 5 years ago, and I think the validity is pretty good now. So if there's a problem, it's with something else probably. You will probably get, depending on the company you go to and what you pay for, you may get some information on disease risk, but you're not going to get the kind of information on disease risk that you would get from a clinical test. And just the one big example of this that gets talked about the most is the 23andMe. I'm not knocking 23andMe. They do a lot of really good stuff, but 23andMe does a test for a couple of locations in the breast cancer gene, but they don't test for all of them. They test for the most common ones. So if you have a positive test for a BRCA mutation from 23andMe, you better get that followed up with a clinical test. But if you have a negative, it does not mean that you don't have a mutation in your BRCA. They're only testing a small subset. Okay. There's a lot of DTC testing that's more what people call recreational. So for traits, for genealogy, for ancestry, it may not matter a whole lot. It may not affect your life a whole lot if you find out that you have a particular variant that makes you more or less able to taste sweet or bitter or salty flavors. Or whether you metabolize caffeine better or worse, or your pee smells when you eat asparagus, or the form of your earlobes, so forth. When you start getting into genealogy and ancestry, you got to understand what you're asking for. So genealogy, some of the folks on the call found new relatives. And if you're good with that, that's great. You might learn that people you thought were your relatives are not your relatives. And this can have huge implications in families. Again, ancestries information is dependent on how good the reference is. And those were not that good. They are getting better. It may not match your family narrative. Most of us are much more admixed than we think we are, and so it's just something to be aware of when you walk in. There are other things. There are potential privacy issues. I'm not sure that there's a lot of hacking going on of these databases, but there have been numerous cases of unexpected use of the data by law enforcement. And some people think that law enforcement should use this information if we can catch criminals. But it can be pretty shocking to find out that your third cousin was the murderer in that county over there. You didn't sign up for that when you put your DNA in. You may have thought that you would have privacy, but the laws are squishy on some of this stuff. There can be insurance risks. I don't know that I've heard examples of this. There are laws against the use of genetic information in employment. There is, as far as I know, there are no, at least nationwide laws that prevent the use of genetic information for adjusting somebody's insurance premiums or insurability. There is potential, and I don't want to end on a downer, so I'm going to end on an upbeat. There's a huge potential for using the information from the millions of people that are collected by these direct-to-consumer companies for drug discovery and understanding health and biology and disease. Again, the consent that you give is key. So again, what I was hoping to elicit in today is maybe a good discussion so that we're all better consumers of DNA information. Realize that all these technologies have upsides and downsides and that we need to walk in with our eyes open. And so I will stop and I'll let Monica moderate.
Awesome. Wow, that was fantastic, Jeff. You really boiled down a boatload of information, or a genome load of information. So yeah, we've got some questions coming in, but I have some. I'm often a fan of the phrases people sort of say, like, explain this to me like I'm 5 years old. This often, the genetics, ever since the Human Genome Project launched, I've often gotten very confused in my head about those different terms. And I'm wondering, can you sort of put in order for me the terms? Like, I get that, base is sort of just that piece of the molecule, but then there's gene, genome, nucleotide, phenotype. And at one point I get kind of lost in where we're talking about this much versus a person.
Okay. So the entire human genome is 6 billion
Right.
of those.
So that's like me, a person, all mine.
That information is divided up, if you will, in 23 chromosomes. Okay. Mm-hmm. Each chromosome contains many, many genes. Mm-hmm. A gene, the classic definition of a gene is it's a string of DNA nucleotides that encodes a protein.
Okay.
It's the instruction book to make a protein.
Okay. So it's
we now include some more. So there's the part of the code that starts at that gives the instruction from the beginning of the protein to the end of the protein.
Okay.
Then there are switches at the beginning of that, generally at the beginning of that sequence. They can be other places that help tell the cell when to turn that gene on to make that protein, to make the RNA that will encode the protein. Mm-hmm. Was that okay? I made a little jump. I threw in
our that helps. And you specifically mentioned that idea of like turning it on, which you hear all the time. And I've also heard the term penetration or penetrating it being expressed. Penetrance of it and expression. Could you talk a little bit about that it's not just having that gene there, which speaks to the testing of something?
So we have roughly 23, I can't remember what's the latest number, 24,000 protein coding genes.
Okay.
24,000 different proteins that our body can code for. Different cells in our body have different proteins. And that's because in some cells, a particular subset of the genes are turned on. In other cells, a different subset of those genes are turned on.
Mm-hmm.
Through development, genes are turning on and off. So there are switches and rheostats, right, to turn up and down the expression, this other term, the expression of each of those genes at the right place in the right time, in development and in just normal biology of your cells. Nerve cells obviously look different, have completely different function from bone cells. That's in part because different genes are being expressed in those different turned on and off.
Okay.
The picture gets way more interesting and complicated because it turns out that in mammalian genomes and most of the higher organisms, there are interruptions in the sequence that codes for the protein. And there are things called introns that have to, as the DNA gets transcribed, copied into RNA, those little introns have to get cut out and the remaining pieces joined together. And now it codes, the RNA codes for the protein. But if you take out different introns in different cells, for example, and join the pieces together, then you have basically the same linear sequence in the DNA that ends up generating two different proteins, slightly different proteins in different cells because of these molecular biology activities that are happening in your cells.
So complicated.
And then when I said rheostats and switches, there's a whole world of study of that. So there's a lot of stuff going on. Penetrance is something very different. Penetrance is when you have a set of variants which give you a more or less chance of having a disease, how much is that expressed in that individual? Mm-hmm. And that's probably controlled by a complex interaction of their genetics and their environment. But the penetrance is referring to a disease. How much is that disease expressed in you?
Okay.
Does that help?
Yes, and that makes sense because where I was hearing that was in a conversation with a friend who's an MD, a doctor in internal medicine. And I was just sort of asking him, do you get a lot of patients getting these tests, coming to you with questions about it, asking about it? One thing I thought was kind of interesting, he said, we're getting less questions than we initially thought. When those, I love that you use the term recreational, the consumer tests. When those first came out, I think the medical community was like, oh my God, patients all over the place are going to be coming, swarming in with these things, very confused about it. As you said, this idea of, oh my gosh, now I've got this disease or that disease, or I'm going to get this. And as he was saying, that's the point that you want to be talking to a genetic counselor.
Exactly.
Because they understand these. And his view was, yeah, they're fine if you get them this way. If you really have a concern, that's why we send you for the real clinical tests. And somebody in the comments was mentioning that. Your internist is going to say, we want to look for this particular thing. And then the genetic counselor can actually share with you what that means. Also, because we're so bad, I included, at that estimating what a percentage increase means in anything. So thank you, that helps a lot. And it goes to one of the questions that came in. Pat Nuccio sort of said, and I wondered this exact same thing. Since everyone has a different genome, how do we say that the human genome has been sequenced? Like, what does that mean? And I remember thinking about that initially. And Craig Vintner was like, he got his done, but what does that really mean?
Yeah, so the communication is really challenging. Mm-hmm. And you have to sell it to Congress and you have to sell it to the public, and that means different things. You have to sell it to different kinds of scientists. There was a huge question at the time whether money should be spent to sequence genomes, human genome. What have we learned from that? Let's study biology. So the first genome, the public Human Genome Project effort sequenced DNA from mostly one individual, but there were 6 donors. I think it was 6 donors. I think they were in Buffalo. I think that's where the library was made. And what we basically have is little overlapping pieces from all 6 of those people. So it's actually not one human. It's pieces of 6 humans, mostly one, but about 6 humans. And actually, the way the sequencing was done, you took a piece from here and a piece from here, from 2 different genomes. And in order to do the sequencing, because of the technology you had to have overlapping pieces. And in that overlap, you might have 2 different individuals. And so they were starting to see the variation in the human genome, even with that first Human Genome Project genome. Okay, but so the Human Genome Project, the 2001-2003 publication, that was, it attempted to be to read across an entire human genome, but it was actually sourced from multiple people. Okay. The competing private sector effort that was run by Craig at Celera sequenced his DNA. Mm-hmm. That was his decision. But that was pretty much that. I think that was one human being. But that was why, we said, okay, well, this is cool, but we know there are differences between people, and we know we're going to need a lot of information. And that, so then, we went on and started sequencing more people. And then, those initial ones were very, very carefully chosen. Well, other than Jim Watson, but they were very carefully chosen. In other words, there'd be particularly when countries wanted to get involved, Korean scientists would sequence 5 Korean individuals, and Han Chinese, there were publications of Han Chinese, and some of the really important samples for many of these genetic studies are from Yoruba or Yoruban, so there are, this was when it really started getting interesting, when we started looking at the genomes of many other people. But the Human Genome Project was it was an attempt to read for the first time most of the information in the genome of the human species.
Mm-hmm. Okay, great. And I think about that because someone I saw give a talk once, your timeline, how fast things moved, and how it shrunk. And somebody said, he said, yeah, in my day, you could get your entire PhD, looking at sequencing one gene, something like that. And now it's done overnight. But that also goes to this point, there's so much talk about the diversity amongst them. But can you explain a little bit more how it gets to, with these new consumer tests, how it finds the similarities to find a relative that I never knew of? Yeah. Like, that has to be so close with the huge variety that must be out there. How do they do that piece of it?
So I'm going to back up a little bit from that, from finding relatives, but I'm going to talk a little bit about the whole idea of how different humans are from each other. Mm-hmm. If we think, there was a concept in Gregor Mendel's genome inheritance studies about random assortment. Everybody probably, if you heard about this in middle school or high school or something, you may have heard that term, random assortment, which is all those little genes can move around randomly. But it turns out the human population is relatively young, and we have what are called haplotype blocks, which just think of big sections of the genome that when DNA is exchanged between chromosomes, which is a normal process, it's in big chunks. And so people from Finland, and I pick on Finland because Finland has done a fantastic job of tracking their populations. They have records back for generations. Okay. And so people in Finland have genomes that are much more similar to each other than people if you compare between Finland and Albania. And part of this is in these large chunks of DNA that just haven't had time to randomly assort. Okay. What you're looking at in close relatives is longer stretches that are similar than in the next degree relative or the next degree relative, or it's just how long is that region that's similar between those two individuals that allows you to identify the family relationships.
So
Because it's not sorting randomly. And so, and the way you identify a block is that there's a bunch of variants. There are not that many, 1 per 1,000. There's some variants. But those two individuals, when you look at that region of DNA, they both have the same variants in there, right?
Okay.
That's how you identify them as being more closely related than someone else.
Wow. And it just blows my mind. That means there are that many people who have conducted these tests so that one of these companies has them in the database to be able to find them and let you know that you have a.
If you get yourself tested and there's nobody else in the database, you're up creek, right? So.
Not related. Okay.
Doesn't mean your relatives aren't out there. They just haven't.
They just haven't done this. Got it. So Al is saying, can you talk about what triggers a gene that can cause a disease, like the gene to turn on? You know, if you have the genes for celiac disease or something else, why does it develop in some and not others?
So, well, the easiest way to say that is the process by which DNA is copied, right? Because in order to make gametes, you have a progenitor cell, and that cell replicates. And now there are two pre-sperm cells or pre-egg cells. And in that process of copying the DNA, sometimes mistakes are made by the enzyme. It's really accurate, but it's not perfect. And so that's a spontaneous mutation. Now, most of the time, what's happening is that these are pretty rare. And so what we generally find with most of the diseases that we have definitions for, which means it's occurring in many people, is that change happened in the individual, in some individual back in time. And that change has been inherited. And depending on various conditions, and those conditions could be other variants in others of your genes, it could be your environment, that disease will be penetrant or not in that individual.
So that's why it's like they're not going to know why two people both have that gene, the inherited gene, why it's turned on, environment.
Well, I mean, it's probably turned on. The gene is probably
on, but not.
The gene is probably needed for some process in your body. And so that's why it's there in the first place, and that's why it gets turned on. And then, I mean, there could be another mutation so that it's not getting turned on in some cells, but it is getting turned on in other cells. And which cells it gets turned on in most would affect whether you have penetrance of the disease. There are so many tweaks to how biology works, which is why it's interesting, and it's also why it's hard to sort it out.
We need more people going into that line of study. Yeah. That's the answer. From Stephanie Call, is most genotyping only concerned with exons? First, you got to explain for everyone else what exons are.
Okay, so the exome sequencing, that was that when I talked about the 5% of the genome that's most interesting. So an exon is a region that's expressed generally as a protein. I could have said that before. There are parts of the genome that are expressed as structural RNAs, as functional RNAs. So there are parts of the genome that get transcribed into RNA that do other things than code for protein. But these are all expressed regions of the genome, and that is exons. You can put together a gene chip to do genotyping out of anything you want to put on that chip. And I believe it's true because it's most of what we understand about the relationship between variants and disease is in protein coding regions or exons. That's because in the 1960s, people figured out the code to, if you have these 3 bases next to each other, that stands for this amino acid. It's just like a translation table, okay? And we have the codons, which are triplet triplets of DNA bases, and the codons code for particular amino acids. And so we've understood that since the 1960s. So that's what people focus on when they put content on a gene chip. So I would say the answer is pretty much yes. It's mostly in the exons. What we're learning now with the ability to sequence more genes, more genomes, and start to interpret that, is that there's all this other information out there outside of the coding regions. And this is mostly where the switches and rheostats are.
Okay.
To control when and where genes get expressed. Those turn out to be really important. And so first of all, a lot of the differences between us and apes are not in what the gene is, but it might be in how many copies of the gene the organism has, and when and where it's expressed, or for how long it's expressed. So these switches and rheostats turn out to be really important. And we have only started learning about them in the past really getting a lot more information about them in the past 5 or so years. So there would have been no point in putting that information onto a gene chip a while ago, because nobody would have been able to interpret it. But now, as we're learning more about these switches, it's possible that more of that kind of information will go on the gene chips, because we're starting to learn how they affect disease, health and disease.
Okay. Okay. And thank you. That may speak to, I'm not sure I understand this person's question entirely. From saying they're data scientists, but not using gene stuff. And he's talking about, if you're looking at, say, Ethiopia to collect data from the Ethiopians and looking for things that it's looking at what I might have in common with them now. But how do you know what was common in them 500 years ago or something? Trying to see what my great grandma, had a similar genome to someone like that. You know what I mean? Is there a way to account for that kind of variation?
So if you sequence the genomes of a bunch of people, you can look, because the same way we were talking about identifying family members by these haplotype blocks, you can look for what's in common among them and what's changed. And so what you will start to see is where there's been admixture, where people from that population started interbreeding with people from somewhere else. But you can sort of use those blocks of information that have not sorted out randomly to turn back the clock and see what they have in common. Now, you have to still be assuming that they were all kind of still geographically located there, because that's the other thing we know in human history: people get displaced.
Yeah.
So it's conceivable that you can look at a location and the people who are living in that location today are completely different from the people who were living in that location 500 years ago, because of the migrations that have happened in human history. That's a different problem. But if you have people who are still actually, the descendants for over 500 years living in the same location, you can actually use the genetic information to dial back to what was dial back. I'm using that in quotes, back to, what was that ancestral population.
Okay. And that's the extent of mine. Many, many years ago, I did a DNA thing for National Geographic, which was tracking worldwide sort of things. And the extent of what I figured out, I'm German and Irish, not uncommon. And shockingly, it said, my ancestors came from Northern Europe. Like, okay, that tells me why I'm a lousy cook. I'm German and Irish. But I think this is fantastic. I'm trying to see if I missed anything on this. I see, as someone points out, it's like, as you're saying, that's like why current Americans, we don't represent Native American DNA, because obviously.
Exactly. That's exactly what we were just talking about. Right?
Yeah. And then you're looking at the cross-pollination of all these different regions now that's going to get more and more, mixed as we want. Yeah,
Homogenized, so yeah.
homogenized, I guess is the way to put it. Yeah, so interesting. I mean, all of this, and I think you're right. As this expands, as it's gotten so much easier to do this testing, and we can only presume that greater technology is going to advance it even further, will provide more and more and more information, not just about, okay, do I have more relatives out there? But as you said, this more precise testing about diseases, which is what really matters. And how can we test for these things, test for them earlier, and then therefore lead to ideally some sorts of treatments or
Right, and you know, I don't want to. We talk about what's happening in the clinic, but of course, you know, this bench to bedside and back, right? The
yeah.
idea that as you look at people who are sick and try to figure out what's going on genetically as well as in their environment and other things, you can start to develop biological insights. So let me just give you one example. There have been huge projects to sequence the DNA from many tumors. And so they'll take all what's considered by the pathologist to be one tumor type, and they'll sequence the tumor and a piece of skin from the same individuals, because you want to look at what's different. What's usually a lot of what's in a tumor is it's a somatic variant. It's a change that's not in that person's genome of their whole body. It's a change that happened in that particular tissue after they were born, after the tissue formed, because of a mutation like skin cancer, right? So what you do is you look at 100 tumors that are the same tumor type, and you look at the tumor and a skin sample from the same individuals, and you sort out what's different between the two. So those are the changes that occurred to make it a tumor. And you look at that for many different kinds of tumors, and then for many tumors of the same kind and many different kinds of tumors. And you start to sort out what genes are being affected. And at first, it's a complete disaster. It's like your head explodes. But then what people are finding out is those are genes in certain pathways that seem to get hit over and over again. And by pathway, I mean like a biochemical pathway, right? And so you start to develop biological insights into the disease. And that's true for the metabolic diseases and cardiovascular diseases too, but it's sort of easiest to explain with cancer. And so from the clinical information that one gets back, you can start to really develop unique and interesting biological insights that give you a completely different, number one, a completely different idea about how to try to attack the disease from a medical perspective. But the other thing it gives you, and this is particularly true in the Mendelian diseases, is that we may have a drug for a particular pathway, but until you know that that patient's mutation is in that pathway, that does you no good. Once you identify what's the mutation that's causing that individual to have that rare disease, very often what's happened is, oh, we actually have a drug in that pathway. Let's try it on that patient. And many times it's working.
And then you learn it. Yeah, which is.
And that's, you could not have guessed from the phenotype. We can go back to that. The phenotype is what's being expressed in the individual. What, when you go to the doctor, here are my symptoms. And you have swelling in your extremities, or you have, that's the phenotype. And most of these diseases are really complex phenotypes. So it's really hard to sort out what's the root cause at a molecular level. But since we can do sequencing now of these people's genomes and newborn genomes and figure out what gene the change is in, it often helps us, gives insight into the pathway. And as I said, often gives us an option to look at a drug that's approved for something else, but might work there.
Which speaks to, we will have to wrap up soon, but so much interesting stuff that you're covering and that people are asking about. Bruce asks, so getting to that point about the complexities of it, do you think that machine learning technology is going to be useful in analyzing this data, like particularly the deep learning idea? Is that going to help?
Yeah, so I mean, I did go to a conference about 3 years ago where several drug companies were trying to use machine learning AI and stuff to try to get insights that the normal human brain can't
Yeah.
derive. And there were huge debates going on about whether this was actually going to be of any use. I mean, I think as it gets better, it's certainly being applied. And yeah, I can't see any way that it's not going to build insights. I've just been involved. I'm retired from NIH, but I do some part-time stuff, and I was just involved in the review of some grant applications where people are using machine learning to try to sort out some of these rheostats and control switches to understand, just the basic networks
Okay.
of how the switches work, and that's going to then expand out to the phenotypes.
Mm-hmm. Okay. I mean, yeah, it makes sense. Like, what else are we going to do with like this insane amount of computing potential?
Right.
I think, but let's apply it to these complicated things.
Yeah, it all depends, as all machine learning, it depends on the training. You got to
Right.
train it right. So.
And who's doing it. This is fantastic. So folks, I do think we better wrap up to respect your time and Jeff's, but very important. So in the chat, if you haven't already seen, Liz, our producer here, managed to insert some of the recommended readings or other information, a NOVA documentary, or not documentary, but a piece about particularly this kind of clinical genetic testing, or the consumer testing rather, and a few other more academic articles with the links to it. So if you want to read more information that he has provided, that is all there for you in the chat. If you want to save the chat, click on the three little buttons on the right, on the bottom there of the chat window. You can do that. And my last little bit, other than to say thank you so much.
Thank you. It's been fun
doing it. Several people have commented in the chat already who had to leave that this has been fascinating and super helpful. So round of applause for you for that, and for dedicating or donating your time tonight to ISC's audience. So folks, remember, for Illinois Science Council, we'll make sure that you get on our mailing list for our newsletter so you can be updated on our upcoming programs and events, including Science of Happiness on the 27th, and our City Urban, not BioBlitz, that effort. And last but not least, I will do a quick share of my screen just to show you what I mean when I'm talking about our vaccinated t-shirts that are now available in our Bonfire shop. I think it's Bonfire/Science Swag. Liz will have that in the chat as well, but there's all kinds of varieties and types of t-shirts in different colors that you can get to when you get your vaccinations, to share with the world that that you have been vaccinated, and show the world and hopefully convince some others to get their vaccine too. As somebody said, so we are recording this, so if you want it for the future, we'll make sure that we share that with everybody on the list today, if anybody couldn't make it who had registered for this. So again, thank you very much. And as we say, stay healthy, stay masked, and be good to your genome. Thanks again, Dr. Schloss. Thank you. Thank you, Liz. Thank you, everyone, for coming. Have a great night.